Hello and good morning. Students, we were discussing about diffusion, and in last class we learned that how a group of molecules which are in a concentrated form, they move and they show a passive movement. So we learned that they go from higher concentration to regions of lower concentration. And I gave you an example of perfume. We find it in our homes, and how when you spray it or open the bottle at one end, you can smell the aroma in the other end as well. So this is done by diffusion, and the gaseous exchange take place, and this is how molecules from higher concentration move along to the regions of lower concentration. But now we are going to see facilitated diffusion. So this is the movement of specific molecules down a concentrated gradient passing through the membrane via specific carrier protein, which means that it is a type of diffusion in which there are proteins. So they only allow specific molecules. For example, here when you see in figure number 1.2, you see the molecules of glucose passing through the membrane into the cell. And how are they doing it? Because they have carrier proteins, and these have a definite shape because they only allow one molecule or same type of molecules which either have the same size, shape or charge. So these, what they do is, these carrier proteins only allow specific molecules, and this is why it is known as facilitated diffusion. Facilitated diffusion means that they provide a special facility to only selected type of molecules that pass through. And this facilitated diffusion is a passive type of diffusion because it does not require energy. Simply there are proteins which have a shape which only allows particular type of molecules to pass, so they require no energy. So it is a type of passive transport, and an active transport is the transport where energy is required. So if the energy inside living organism is required for something to move, and that energy is ATP, adenosine triphosphate, so then we will call it active transport. So I believe you understood this point. Facilitated diffusion, it means that generating a facility in a membrane where proteins or carrier proteins are used for specific type of molecules which either have same size, same shape or charge. So these are the common molecules entering or leaving cells. Alright, now we see osmosis. So what is osmosis? It is a special example of diffusion, and it is the diffusion of water through a partially permeable membrane from a more dilute solution to a more concentrated solution. So we saw in diffusion that it goes from a higher concentration to a lower concentration, but in this it will go from a lower concentration, here it's lower concentration, to a higher concentration. You see these dots which are molecules of salt. So in this part they are less, but here they are more. But water will move along from this area to this area, and this type of transport is known as osmosis. And we see that they move along because they down the water potential gradient. Okay, so from permeable membrane, which means which allows the movement. And in this diagram we can clearly see how this example is showing the process of osmosis. So in this, a partially permeable membrane is a barrier, so it stops, but it at the same time allows or permits the passage of some substances, not others. So here water is moving along this concentration gradient. So osmosis is when diffusion of water takes place from a region of lower concentration to a region of higher concentration. So they move along the cell membrane. And in this we see that the presence of solutes stimulates the membrane to open specific channels or trigger active transport. And we can see some examples also, like absorption of water by plant roots. So plant has a higher concentration inside the cells of the plant, but they absorb water from the soil. And here reabsorption of water by proximal and distal convoluted tubules of the nephron. So nephrons are found in the kidneys, and what they do is reabsorb the water. Whenever we have less water in our body, then the water which is being filtered, the nephrons, they reutilize, because nephrons have inside the blood more concentration, but they absorb water from a lower concentration, but the water concentration is higher. Next point is reabsorption of tissue fluid in the venule ends of the blood capillaries. So in blood capillaries also, these capillaries, they absorb excess of water again. And in alimentary canal, it is the absorption of water in our stomach and small intestine. Next we will see osmoregulation. So osmoregulation is keeping the concentration of cell cytoplasm or blood at a suitable concentration, which means that it is the regulation of osmosis. So by osmosis, our body regulates the concentration. Inside the cell, there is cytoplasm, and there it maintains the level of water, because even higher concentration of water can be harmful to us. So this is why an examples have been given for amoeba. They live in fresh water and they use a contractile vacuole. So what these vacuoles, these empty spaces, what they do is they expel the excess water. They just leave the excess water in the water itself. And even in kidneys, which maintain the blood concentration. So this is how osmoregulation helps a body to maintain internal environment. Now let's see osmosis in our plant cells. So we know that plant cells in a hypertonic, which means weaker solution where the concentration is less, have lower water potential because they cannot absorb much water. So hypertonic solution is a solution where the concentration is weaker or less number of solutes, and the plant cell gain water by osmosis. And we know that the vacuole and cytoplasm increase in volume, so it expands, and we are going to look at this example. So now look at this figure 1.4. Here, see, there's a wall, and this shows a cell. A vacuole is inside, and this is a plant cell, and the yellow part that you see is the cytoplasm. So when it is placed into a solution with lower water potential, example concentrated sugar solution, then cell content will become like this, because the excess of water will go out. But when it is kept in a solution with higher water potential, which means a dilute sugar solution of pure water, then the cell content will become like this. So this form of cell is known as turgid, and this will be known as flaccid. So kept in concentrated solution, the cell will become like this. The water will go out, and here, if it is kept in weaker solution where water is more, then water will be pushed in, and the cell will become turgid. And we can see this in these lines on page number 20, that the cell membrane is pushed harder against the cell wall, causing it to stretch a little, and the plant tissue becomes stiffer. So solution. But when it is kept in higher solution or hypertonic, then what happens is plant cell lose water by osmosis, and vacuole and cytoplasm decrease in volume, and cell shrinks, and shrinkage only stops when the cell is at the same concentration. And this example you can see when you place a grape in a sugar solution, or when you place a gram in water solution. So we know that the plant tissue becomes flaccid, and it has shrunk slightly, so it may go on to become a plasmolysed cell. And in next class we will learn about what is turgor pressure and active transport.